Hi, my name is Dr. Patrick Fries. I'm a soil health scientist here at Ward Lab. I focus a lot on soil health testing, method development, data analysis, and customer support. What I would like to talk about today is active carbon. So we had some previous videos looking at these different carbon pools in the soil, organic carbon pools, active and stabilized or long-term carbon, that we quantify using just a simple sieve method. We also have other methods that look at this active carbon fraction in the soil. And why this pool is so important is that this is really the fuel for microbes in the soil. Especially if you want to build long-term carbon storage, you need a lot of fuel to build up microbial biomass that goes into long-term carbon production. But today we have a lot of different methods at looking at active carbon in the soil. So I thought it would be good to kind of look at how these, what these tests are and how they kind of relate to each other, because it can be a little bit confusing, especially if you're looking to measure your different organic carbon pools in the soil. So some of the different pools that we have or that we look at are particulate organic matter, and then also water extractable organic carbon, permanganate oxidizable carbon, and then also mineralizable carbon. So we have four, broad tests that we can do to look at your active carbon pools in the soil. So looking at the broadest or the largest pool of these is particulate organic matter. In our previous video, we looked at a sieve method for quantifying this amount of particulate organic matter, a palm fraction in the soil. And how we do this is the 53 micron mesh sieve, which is generally the silt and clay fraction of the soil. And we take a soil, we shake it with a certain solution overnight, and then we put it through this mesh sieve. Anything that sits on top of the mesh sieve is the active carbon fraction, the palm fraction. And that's mostly comprised or made up of plant material that the microbes use for fuel. That's one of the largest active carbon pools in the soil compared to water extractable and then POC or permanganate oxidizable carbon. It can be roughly about 20% of the organic carbon in the soil, so it's quite large. So the particulate organic matter or palm fraction generally makes up about 20% of the organic matter, organic carbon pool of organic matter, so it's quite a significant amount. The next one is POC or permanganate oxidizable carbon, and again this uses a somewhat aggressive oxidant, but it's a very quick test. So it's a short reaction that we do just to look at that very reactive active carbon pool in the soil. This makes up about 20% of the palm fraction. So it's just a few percent of the organic matter, organic carbon pool as a whole. It's kind of confusing keeping track of all these different pools, but kind of relating these to each other is really important to see what you're actually measuring in terms of how active that fraction is. The next one is the water extractable organic carbon. This is actually what we test with our soil health assessment. This is probably the most active fraction and relates directly to respiration, especially in our reporting of this amount. The water extractable carbon, organic carbon, is what we just pull out passively with water solution. And this is about probably about 1% of the organic carbon pool of the soil organic matter. So it's a very, very small fraction of the organic carbon that we look at. This is directly the food for microbes that they use of the active pool, which goes into their general activity and building up organic matter, quality organic matter in the soil and long-term carbon in the soil. So these are some of the different pools that we look at. There's also mineralizable carbon, which is just a 24-hour incubation that we take with a small amount of soil, we wet it, and we incubate it overnight. We measure the CO2 coming off, and that directly relates to general microbial activity, which is sort of the engine of the system. We're looking to see how active those microbes are. If you have really high active respiration, high activity in the soil, if it's too high, this can be bad because carbon can be limiting in the system. You want sort of a normal steady state, nothing too high. We have ranges for these for our reports. So you want to sort of hit a nice mark. All these pools are facilitated by residues, root exudates, rooting systems, diverse rooting systems, which help build up a lot of this active carbon and goes into microbial activity and then eventually quality organic matter and long-term carbon storage in the soil.